Good evening, friends. Today we will be discussing in this video nucleic acid hybridization. Now, when I am talking of nucleic acid hybridization, what does it mean? It means that it is a methodology or it is a tool which takes the advantage of individual single-stranded nucleic acid which are complementary to each other to be paired or hybridized. Now, this particular hybridization depends upon one of the very important factor, that is your degree of complementarity, that is how many base pairs are actually complementary to each other, that will dictate the efficiency of your nucleic acid hybridization. Now, nucleic acid hybridization can produce hybrids of DNA. That means you may get a DNA hybrid, or you may also get two RNA hybrids, RNA hybrids, or a you can say a heterohybrid, that is your DNA RNA hybrid. So your hybrids can be either DNA RNA hybrid, or your DNA RNA hybrid. Now the question is that why do we require hybridization? We require hybridization to find out certain genome with specific target genes. We can get certain target gene. We can find certain target gene by development of a complementary, by development or developing a complementary oligonucleotide, complementary oligonucleotide, and this is what is called as, in one word, what it is called? It is called as probe. It is called as probe, a short oligonucleotide strand which is complementary to the sequence of your target gene. So this probe will help you to search the target gene in the entire genome. So if you want to search a target gene, find the target gene, we require to produce a complementary oligonucleotide probe. Now, your next point of discussion is your stringency. Stringency of hybridization reaction. What do you mean by your stringency of hybridization? What is stringency? Stringency means specificity, perfection. Now, stringency in hybridization describes that number of mismatch bases that are allowed in hybridization reaction. That means what? Suppose there is a hybridization reaction going on, like ATTGGC, and you have C, A, C, G, C, C, G. So here you get to see this is complementary, this is complementary, this is complementary, this is complementary, but your TC is a mismatch. So how many such mismatch is actually required, or not required, which are allowed in a particular hybridization reaction? That is your stringency. That means if the stringency is more, then there will be low level of mismatch, low number of mismatch, low mismatch. If the stringency is very high, it will have very low level of mismatch, and if your stringency is low, then there will be high level of mismatch, which is not at all desirable in your particular hybridization reaction. So what are the factors on which the stringency depends? So the factors on which stringency depends. Stringency depends basically on two factors. What is that? First is your temperature, and second one is your salt concentration. These are the very, very important factors. Now, temperature means that high temperature, high stringency condition is always achieved with high temperature. That means if your temperature is high, and it is approaching your Tm. What is your Tm? Tm is your melting temperature. Tm is your melting temperature. So if it is reaching your melting temperature, then your stringency should be high. But if you have low salt concentration, so this is high, and if you have low salt concentration, then again if the salt concentration is low, then you have a higher stringency. So these are the two factors on which actually the stringency depends. So next thing is that how this temperature and salt concentration is related to your stringency. Now you know that for optimum hybridization temperature, your optimum hybridization temperature should be 5 degrees centigrade lower than Tm. What is Tm? Tm is the melting temperature. That is the temperature in which 50% of the complementary strands are separated. So basically your particular temperature, that is your starting temperature, should be 5 degree low for optimal hybridization. But when I am talking of your salt concentration, you know that your DNA is having a net negative charge. That means it is having phosphate backbone. Now this phosphate backbone is having a net negative charge. Now this particular net negative charge, this is a net negative charge. Another DNA, suppose this is your target DNA, this is your target DNA, and this particular target DNA is having your phosphate backbone with the negative charge. Now this is the particular probe, and this probe will also have your phosphate backbone with your negative charge. So what will happen? There will be a repulsion. But if there is a repulsion, how hybridization is possible? Hybridization is possible because your solution is having salt, and that salt is a cation. So the cation will actually mask the charge. The cation will mask the charge, one of the charge, maybe the charge of the phosphate, and will allow another DNA to be hybridized. The repulsion is nullified by addition of cations. Addition of cations. But when your salt concentration is low, what will happen? This masking effect will not occur. This masking effect will not occur. That means negative charge will persist and your repulsion will persist. And as a result, what will happen? More amount of mismatch can be more amount of mismatch since it is actually your lowering of salt concentration. So what will happen? This lowering salt concentration, the DNA solution promotes denaturation by removing the cation that shield the negative charge on the two strands from each other. So at low ionic strength, the mutually repulsive force of this negative charge from phosphoryl group are enough to denature the DNA, even at a relative low temperature. So at low concentration, the two strands can only stay together. These two strands can only stay together if and only the most of the bases are complementary to each other. If it is not complementary to each other, if it is having mismatch in it, it will be simply separated, because the positive cations are removed, only the negative ions are there, so there will be always a repulsion naturally. So that is why it says that stringency of a particular hybridization reaction is promoted by a high temperature and your low salt concentration. Next is nucleic acid probe. So, for your nucleic acid hybridization, one of the very important aspect is your nucleic acid probe. Now you know that this particular nucleic acid probe can be a DNA probe, it can be a DNA probe, or it can be a RNA probe. So it can be a DNA probe or it can be a RNA probe, and it is basically made up of oligonucleotide. The minimum 20 1 minute. Sorry for the technical disturbance. Minimum 20 nucleotides are required for the probe.